Hi, my name is Sarah Liner. I'm the Chief Executive of Fios Genomics. I'm just going to give you a very top level overview of Fios, who we are, and what we do. So, Fios Genomics is a University of Edinburgh spin-out, and the company was formed back in 2008. And so this year we entered our teenage years. We're based in Scotland in an area called the Bio Quarter in the city of Edinburgh, Scotland's capital. Although, like many companies, the team is currently working remotely. Fios is a bioinformatics service provider. We don't have any laboratories, we're not involved directly in the generation of data. However, if that is something our clients need, we can organise that through a network of third party laboratories. Our clients are predominantly pharmaceutical companies, biotechs, data generating contract research organisations, and we still do some work in the academic space. Within these groups of clients, there's actually a really wide range within each of the companies. We work with biologists within each of these groups, and then we also work with the bioinformatic departments of these groups, serving as additional overspill capacity or an additional set of expertise where required. Fios Genomics has 45 employees, but despite our relatively small size, we've completed over 600 contracts in the years that we've been going. Fios has been partnered with Charles River Laboratories since January of 2020, and through them we can also offer access to a wide range of data generation assays and also in other areas of preclinical development if required. But why does Fios exist? There have been huge advances in the data generation techniques over the last decade, and it's opened up whole new avenues for development and process improvement. Things like biomarker identification, the stratification of patients within a clinical trial, the field of personalised medicine, and also in the development of biologicals. These subject areas have been really advanced in this big data revolution. And that was the main reason that Fios was formed. But with a lot of the good stuff comes some issues. And these are just some of the issues that our clients commonly have. Starting with something quite simple, the volume of data. Now, big data means different things to different people. My interpretation of big data is just data that you can't readily extract the information from. But if we start with the basics, the volume of data can be a really basic issue for people. Moving data around, getting it from where it's been generated to where it needs to be analysed. Very basic issue in 2021, but something that we see quite commonly with our clients. And equally, there's a diverse range of outputs that people can have from different platforms. So you might have to switch platforms because of a technology advance. You might have to move from labeled to unlabeled mass spec, for example. So this again can cause difficulties when you're used to working with a particular data type. There's a lack of gold standard in the regulations for what's required for genomics analysis. Now, we know our work has been in front of the FDA. However, clients are very much flying blind as to what analysis to do for what data type and where. What technology to use and when is a difficult question that some of our clients have. And when I say technology, I mean, is it gene expression? Is it genotyping? Is it proteomics, for example? And this is where I introduce this jigsaw analogy. No one technology is going to give you the entire picture of what's going on in your experiment or your trial. But which technology is the corner piece of that jigsaw that's going to allow you to build up that data picture of what's happening? And then last, but probably the most relevant reason that Fios was created was the interpretation issues. What is this data telling you? What does it mean? What is it telling you about your drug, about your model, about your patients, or about the disease that you're studying? And that was the main reason that Fios came into existence. In terms of the services we offer, it's quite varied. It is nice to get involved as early in the process as possible in terms of things like experimental design. People think about bioinformatics as being at the end of the process. However, a discussion with a bioinformatician can make sure that your study is controlled adequately, that it's powered adequately, that you're not introducing bias inadvertently by your study design or the way that you're going to profile the samples, etc. And these are just things that can be discussed in a simple phone call with the Fios team. Now, Fios is not a software provider. It's important to stress that. We're very much a consultative service provider. It's not an off-the-shelf product because every data set is different and every client is different as well. So instead, we have proprietary workflows that we use in-house that knit together commercially available and academic tools depending on the data type and the type of analysis that you're looking for. So it's very much bespoke and consultative. We can help with the technology and platform selection. As I mentioned, we don't have labs at Fios ourselves. However, we can arrange the data generation through a network of third-party labs that we know and trust, and that also includes Charles River, who are our partner. Fios is completely platform agnostic. It genuinely doesn't matter to us how or on what platform the data has been generated. We've come across most of the commercial platforms. On occasion, if it's one we haven't seen, we may ask for an example data output just to make sure that we can analyze that data. Just a note on data, as well as data that you are generating, Fios also offers some services in public domain data. And that can start with data landscaping, data sourcing, looking out to see what data that you're interested in is available in the public domain, either in publication or some of the big data repositories. So we can go data hunting for you and then perform the analysis of that data with your question in mind, rather than that of the people who generated the data. One of the key sets of skills that we've developed at Fios is our ability to integrate omics data with other quantitative outputs. So think of a clinical trial. You may have RNA-seq or genotyping data from your patients, but you're also likely to have efficacy data. You might have imaging data. You might have blood data or FACS analysis data. You'll certainly have patient metadata, patient demographic information. And what Fios does is layer up those pieces of information and analyze them as a single data set rather than separately in silos. And that allows you to see quite clearly if there's patterns or trends or clusters within your data set. Some of the data types that we work with are listed here in the diagram below, although this list is not exhaustive. Just to run through a very top level example of how we approach analysis, and I'm using RNA-seq as an example here, but really it can be any data type. We do like to get data in its rawest form at Fios, either if that's data you have generated, or a third-party contract research company, or indeed data in the public domain. And that just means that if there's any particular issue in the data, we have the raw data to go back to check what potential errors have been introduced. That being said, we can accept data slightly further down the pre-processing route if need be. The first thing that we do with any data set, regardless of source, is a QC and a normalization process. We firmly believe that if you put rubbish in, you're going to get rubbish out. And so this QC stage is vital to the overall success of a particular project. I always say to our clients that if you can ignore the QC section in your report, it's a good thing. If any issues come up in this part of the analysis, we will stop and we'll organise a discussion with you to see how you would like to proceed. The last thing that we want to do at Fios is overfit data or have you making decisions or drawing conclusions from data that isn't of robust quality. The next thing with a RNA-seq study would be to generate a gene list. So a list of genes that are differentially expressed in the comparisons of interest. Now, this is where a lot of the online software solutions will stop. This is where a lot of the data generation providers will stop in their analysis. And despite the fact I'm a PhD qualified scientist, a gene list doesn't really mean much to me. It's just more data. But at Fios, we're all about turning that data into information. And so we always take the data to the next level. And we do that by looking at things like network and pathway analysis. What is the pattern of coordinated gene expression, in this example, from that gene list? So what pathways are being impacted? Or is it very much a kind of scattergun type approach to the effect that your drug or your disease is having? And this is where our reporting infrastructure really comes into its own. So our reports are provided as HTML links. They are searchable, they are clickable, they are dynamic in their architecture. And that allows our clients to interrogate and explore their data without necessarily having any bioinformatics knowledge of our own. And we can also talk you through those reports to help you get the most out of it. The next stage in the analysis is very project driven. So we could be integrating other pieces of the jigsaw puzzle. Do you have efficacy data? Do you have patient information? Anything else that can help build up the picture of what's happening? And then we'll perform things like clustering, how are samples grouping together? For example, could we identify patients that were not going to respond from baseline based on some baseline markers? That's just one example. We could then pull in other types of data, public domain data, look to see how things are comparing. And then the key thing, as I mentioned before, is the biological interpretation. What does it all mean? And from that, we use our expertise that we have gained from the 700 plus studies that we have done. And we understand biomarkers. We understand red herring biomarkers that always come up in disease states. So very much about making sure we get the right biomarkers. And then the visualization is another key strength at Fios. It's more than just heat maps. And again, the dynamic nature of our reports really helps here. As I mentioned, we're completely platform agnostic, so we can work with all of the data types here, regardless of how they have been generated. We're also therapeutic area agnostic. So a lot of our work is in the oncology space. However, we've done a lot of work in dermatology, cardiovascular research, GI and microbiome, which are really booming at the moment. A lot in vaccines, as you can imagine, and then respiratory. But we have worked across a great deal more, but these are indeed just the main ones. So really, just to summarise, Fios Genomics is a bioinformatics service provider providing platform-agnostic analysis services to pharma, biotechs, CROs, and some academic institutions as well. There's a variety of ways that people work with Fios, either through a per-project basis or on a full-time equivalent basis, and that's very much driven by the client, and we're happy to discuss the best options. If you'd like some more information, please don't hesitate to reach out. There's also quite a lot on our website, fiosgenomics.com. Thanks.